Okay, cool. I guess we'll get started here. Uh, Hi, everyone. Thanks for coming to this week's BGSA seminar. Hope you guys had a good Thanksgiving break. This week, we're lucky enough to have Dr. Ashlyn Swift-Gallant from the Department of Psychology. If you could please mute your microphones till the end of the talk, and the question period will follow at the end of the talk. And with that, we would like to respectfully acknowledge the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We also recognize the Inuit of Nunatsiavut and Nunatuavut and the Innu of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful and equitable partnerships with all the people of this province as we work towards true reconciliation and honor this beautiful land together. Yeah, and if you're ready, Ashlyn, go ahead and take it away.
Great. Thank you, Cole, and thank you for inviting me here today. I'm excited to share some of our research with you. So overall, our lab is interested with how the processes of sexual differentiation shape the brain and behavior. And today I'm going to present some of our research looking at how hormones influence sexual preferences and where hormones are acting to do so. So in the talk today, I'm going to begin with a brief overview of the field to provide the framework for the studies that I'm going to present. I will then present some of our research investigating site of action. So in these studies, we ask where hormones are acting to affect behavior. And I'll discuss some of our research showing that not all of the action is happening in the brain, but hormones outside of the brain can also have a profound effect on behavior. I'm then going to discuss how some of our research from the rodent work, how that translates to humans. So I'm excited to share this with you. So back in the 1950s, Young and colleagues first described how sex hormones in early development can affect sexual behaviors expressed in adulthood. So typically, males are exposed to high levels of androgens like testosterone that's produced by their testes. And in adulthood, these males have a preference for females and they actively attempt to mate with females. But if we remove the testes of males during these early developmental periods, then males will no longer exhibit a preference for females and their behaviors will tend to be more female typical in adulthood. On the other hand, females are exposed to low levels of androgens like testosterone in early development. And in adulthood, they're more likely to show female typical sexual behaviors like lordosis. But if we expose females to male levels of testosterone, then females will display more male typical behaviors as adults. So this landmark study taught us two important principles that continue to hold true today. So testosterone is largely responsible for sex differences in behavior, and the presence of hormones in early critical periods can have long-lasting effects on behavior. So these are known as organizational effects. So in this landmark study, Young and colleagues hypothesized that hormones are acting in neural tissue to affect sexual behavior. And although this might seem obvious to us today, this was truly revolutionary to the field, and many to this date continue to study how hormones act in the brain to affect behavior. And there is now a great deal of evidence to support a role for hormones in shaping sex differences in the brain. And similar to behavior, these sex differences are highly dependent on the presence of testosterone. So in other words, exposure to high testosterone during early development tends to lead to a more male typical pattern in the brain, whereas the lack of testosterone leads to a more female typical brain development. So many of these sex differences are in the accessory-olfactory pathway. So these brain regions are known to play key roles in sexual and aggressive behaviors. So in the brain, we have the accessory-olfactory bulb, which is attached to the main olfactory bulb. I don't know if you can see my cursor, but I'll point that out. And downstream from there, we have the bed nucleus of stria terminalis, the medial amygdala, the hypothalamus. And so these brain regions are receive input from the vomeronasal nasal organ, which is in the nasal cavity, and it is responsible for sensing pheromones. And so it sends information about conspecifics to this neural circuit. So most research to date has supported this framework by Young and colleagues, namely that hormones act in the brain to affect behavior. But few have considered the alternate hypothesis. So during the time that Young colleagues proposed this theory that hormones are acting in the brain, another principal founder of the field, Frank Beach, proposed that the more parsimonious explanation would be that hormones act on the genitals to affect sexual behavior. So this idea can seem strange at first, since we're so used to thinking about the brain being responsible for behavior, at least in psychology. But Beach explained that having high testosterone during development leads to the growth of a phallus in males, and because males have a phallus, they have a means to penetrate. So males are more likely to show mounting and thrusting because they can. During this time, Beach himself did not have the tools to directly test this hypothesis, but we do now have interesting genetic tools to test this idea. So for example, with new genetic tools, we can alter hormone sensitivity in neural versus non-neural tissue by manipulating the androgen receptor. So androgens like testosterone can exert their effects by acting on the androgen receptor. So androgen receptors reside in the cytoplasm of the cell, and when androgens are present, they act upon it, and the androgen receptor translocates to the nucleus and affects gene expression. So if we're manipulating the amount of receptors available in neural versus non-neural tissue, we can address this question of whether hormone action in non-neural tissue affects sexual behavior or sexual preferences. So there are multiple strategies we can take with manipulating receptors. We chose a strategy of overexpression because this not only allowed us to ask questions about site of action, but it also allowed us to ask how behaviors change in response to high levels of testosterone. So we did this using the Cree-LOXP system. So the androgen receptor transgene that we used was present in all cells, so both in neural and non-neural cells, but as you can see, it has a stop sequence. So it needs to have Cree recombinase for the stop sequence to be excised. So we can manipulate the functioning of this transgenic receptor by manipulating where Cree is expressed. So we made two models. So we had one model where we expressed Cree in all tissues, and so it resulted in a model with global overexpression of androgen receptors. And for a second model, we used nestin Cree, which is a neural-specific Cree to overexpress androgen receptors in neural tissue. So the important distinction between these models is that one model has more androgen receptors in neural tissue and the other one has more globally in both neural and non-neural tissue. So if we are to find that both models show a difference in a behavior, then we can deduce that neural androgen receptors alone are sufficient to affect that behavior. But if we only find differences with global overexpression, then we know that neural AR was not sufficient to affect behavior, but we also needed expression in non-neural tissue. So the principal questions we wanted to ask here was, do androgen receptors in neural tissue contribute to sex differences in behavior? And is there also a role for non-neural androgen receptors? And we also asked if androgen receptor overexpression can lead to an increase in male typical behavior. So to address these questions, we looked at various measures, and I'm just going to focus on a couple today that relate to sexual preferences. So let me just turn this down so you can hear me. So one test that we used was the olfactory preference test. So you can see this here. So we'll expose them to male soil bedding, female soil bedding, and a clean stimuli, like so clean bedding. And so we'll see how much time they spend with each stimulus, and we'll calculate a preference score so we can see how much time they spend in female bedding versus male bedding. So typically males love female bedding. Next, we also exposed males to female soil bedding prior to dissection. And so this allowed us to look at a marker of neural activity in the brain. So we looked at FOSS. So you can see here, typically when a male is exposed to female soil bedding, they'll show an increase in activation in brain regions along the accessory olfactory pathway. Whereas if they're exposed to clean bedding, there won't be very much activation. So here I have plotted the olfactory preference results. So a positive score here indicates a preference for female odors, whereas a negative score will indicate a preference for male odors. So as you can see from this graph, if we look at wild type males, they have a preference for female odors compared to females wild types. We found the same pattern with mice with neural overexpression, but when androgen receptors were overexpressed globally, we found that there was a decrease in preference for female odors. So the males here actually had an equal preference for both same-sex odors and female odors. So this suggests that global androgen receptors, but not neural androgen receptors, are affecting sexual preferences. So next we looked at neural activity along the accessory olfactory pathway. So we looked at the nucleus accumbens, the medial preoptic area, the bed nucleus of the stria terminalis, the medial amygdala posterior dorsal division, and the ventral medial hypothalamus. And across the board, we found that global overexpression of androgen receptors led to a decrease in neural activity compared to both wild type mice and mice with neural only overexpression. So these results correspond with the behavioral results. So we're seeing a decreased preference for female odors and a decrease in neural activity in response to female odors. So these results, together with other experiments that are published, so I won't go into much detail about today, indicated to us that androgens can act in non-neural tissues to affect the sexual differentiation of behaviors. And we found this unexpected finding for this behavior as well as others, that at the high extent of androgen signaling, male mice show an increased preference for same-sex odors. So I found both of these findings particularly interesting, and we have pursued separate lines of investigation from both. So first, we asked what non-neural targets were involved in sexual preferences. So obviously, one of the first things we thought of was thinking back to Frank Beach. Well, perhaps the genitals are one non-neural peripheral site of action. So it is possible that with global overexpression, increased genitalia size or increased sensitivity of the genitals could increase the rewarding effects of mounting and thrusting, for example. But for other behaviors like sexual preferences, it was unclear to us how the increase in sensitivity of the genitalia could affect behavior. So another one that we thought might be interesting is the vomeronasal nasal organ. So as I mentioned before, this organ sits in the nasal cavity of mice and is responsible for processing pheromones. So it has both neural and non-neural cell types, and it speaks directly with the accessory olfactory bulb, making it an ideal candidate. So I will discuss some of our research today, asking whether the vomeronasal nasal organ is a site of action. In addition to the VNO, we're also interested in studying the gut microbiome and how it interacts with the endocrine system to affect social sexual behaviors. And our preliminary results do suggest that depletion of the microbiome can decrease androgens and decrease male territorial aggression. But I don't have enough time to go into all of this today, so I'm going to focus on some of the VNO work. So a lot of what we know about the role of the VNO comes from ablation studies. So more recent studies have used genetics to impair the vomeronasal organ by knocking out the TRPC2 gene. So TRPC2 is a calcium ion channel highly expressed in the vomeronasal organ. And when this gene's knocked out, the cells in the vomeronasal organ cannot communicate information about pheromones to the brain. So interestingly, these mice can still discriminate between males and females, and that's likely due to their mating olfactory system. But sexual preferences are altered. So for example, males will show an equal preference for mounting male and female conspecifics. And these males also show a decrease in territorial aggression. And similarly, females show sexual behavior towards both same-sex and opposite-sex conspecifics. But what is particularly striking is the females are exhibiting male-typical behaviors like mounting and thrusting. So these very androgen-dependent behaviors are increased in female mice from this knockout. So we had a couple questions in response to the VNO research. So with genetic models, such as the TRPC2 knockout, it results in a lifelong ablation of the vomeronasal organ. And as we've discussed, many social sexual behaviors are dependent upon early critical periods in development, like the day of birth and puberty. So we've asked if adult ablation is sufficient to affect social sexual behavior, or if pubertal ablation would similarly affect social sexual behavior. And since these behaviors that are affected are androgen dependent, we were also interested in whether androgen action in the VNO could mediate these sex typical behaviors. So first, to address the organizational effect of the VNO, we did surgical ablation. So we either ablated the VNO at pubertal onset or in adulthood. And we compared these animals to sham-operated controls. And we looked at a number of tests, including the buried food test, which, as it sounds, it's a test in which we bury a food stimulus, like a cookie or a Fruit Loop, and we see whether the mouse can smell, so if they can uncover that food stimulus. We also did the olfactory preference test, which I've already discussed. And we also did a resident intruder test. So in this test, we presented experimental animals with a stimulus mouse into their home cage. So typically, a male exposed to an intruder male will aggress. And conversely, if we present them with a female stimulus mouse that's in estrus into their home cage, the male mouse will typically show sexual behaviors towards her. So first up, we wanted to make sure that our animals could still smell. And so we looked at the buried food test. And as you can see here, we did not find any statistical differences between our groups. So all animals were able to recover the food in the allotted time. Then we looked at the olfactory preference results. So again, a positive score indicates a preference for females and a negative score indicates a preference for male odors. And we found an interesting finding here with the male mice that had pubertal ablation. So we found that they had a significant decrease compared to sham-operated males, and adult VNO ablation mice were intermediate, such that they didn't differ from shams or the pubertal VNO ablation. We didn't find any differences between our female groups. Next, when we looked at the intruder test, so we in response to a male intruder, we can see that overall there's a sex difference where females are investigating the face and body of the intruder more than males. And we found that pubertal ablation, males with pubertal ablation, showed an increase in investigations. They're sniffing the body and the face of the male intruder more. And if we look at aggressive behaviors, we see across the board that there is a decrease in aggression in male mice with pubertal ablation, but male mice with adult ablation continued to show aggression similar to the sham-operated controls. So first up, we had latency to aggress. So the pubertal ablation, males are showing an increased time before they show any aggression. And overall, they're showing a decrease in chasing and a decrease in attacks. So attacks here was defined as pouncing on the male intruder. And I don't have here the graphs for sexual behavior, primarily because we did not see any differences in sexual behavior. So all males showed sexual behavior towards a female intruder, but there were no differences by VNO ablation condition. But we did find differences in neural activity as approximated by FOSS in response to opposite sex odors. So here males were exposed to a female odor stimulus and males were exposed to, so males were exposed to a female odor and females were exposed to male odor. And again, we looked at the same 5 brain regions in the accessory olfactory pathway. And as you can see here, we found that both male and female mice with a pubertal ablation of the VNO showed a decrease in neural activity compared to both mice with adult ablation and mice with sham-operated controls. So to summarize these findings, these results suggest that that pubertal but not adult VNO ablation alters territorial aggression. So this suggests that the VNO during puberty is contributing to the organization of the neural circuitry underlying these behaviors. We did not find any differences in sexual behavior, but we did find differences in sexual odor preference and in neural activity in response to opposite sex odors. So this suggests again that pubertal but not adult ablation are affecting these measures. But we did not find any differences in sexual behavior, which was surprising. So mice with lifelong ablation via genetic tools, so like the TRPC2 knockouts, they showed drastic differences in their sexual behavior. But pubertal and adult ablation was not sufficient to affect sexual behavior. So to us, this suggests that the early critical period, so around the time of birth, is probably an important time for the VNO functioning to affect sexual behavior. Unfortunately, we were not able to test this with a surgical ablation method, as neonates with the VNO surgically ablated did not survive. So next we were interested in the hormones. So can androgens act in the VNO to masculinize behavior? So for our initial studies, we just did a local micro injection of testosterone or an injection of a vehicle, so just oil, into the vermeil nasal organ on the day of birth. And we looked at social sexual behavior in adulthood. And so we did all the same tests, and what stood out were that both sexual and aggressive behaviors were altered. So testosterone in the VNO on the day of birth was sufficient to increase territorial aggression, but it decreased sexual behavior such as mounting. So what we have so far suggests that puberty is a critical period for VNO mediation of aggression. And based on the TRPC2 knockout experiments in comparison to our findings, it would indicate that the early critical period is likely an important time for the VNO mediation of sexual behaviors. And our preliminary results suggest that testosterone in the VNO is affecting territorial aggression and sexual behavior. So currently we're doing some studies where we're targeting the androgen receptor or the estrogen receptor. So testosterone can convert to estrogen and work on estrogen receptors. So we're trying to figure out which receptor in the VNO testosterone could be acting upon. So we're currently doing this with antagonists And once we figure out which receptor is likely involved, we are excited to try maybe viral vectors or transgenic techniques to target the sex hormone receptor. We're also interested in bringing some of this together, and we wonder whether testosterone in the VNO at puberty could masculinize aggression. So if that's the time, given that ablation at puberty is affecting aggression, perhaps hormones during puberty are affecting aggression. And finally, we're excited to look into the brains and see how these vomeronasal organ manipulations are affecting the downstream neural targets. So in addition to these studies pursuing this non-neural site of action, the second finding that high androgen signaling can increase same-sex preferences in mice led to a separate line of investigation in humans evaluating the translational value of this work. So specifically, based on these findings, I asked whether it's possible that high androgens can lead to same-sex sexual orientation in humans. And if we look at the literature, it's quite mixed. So there's some studies that indicate that gay men compared to straight men might have an increase in androgen. Others indicate low androgen. Many others report no relationship. And so this led me to ask whether there may be subgroups who differ in their biological mechanisms that contribute to their same-sex sexual orientation. So for example, perhaps some gay men are exposed to high androgens, whereas others are exposed to low androgens, which could account for these conflicting findings. So to test this idea, we used anal sex roll as a proxy for subgroups in our initial studies. So anal sex roll refers to the sexual position that a gay man takes during anal sex. So bottom refers to gay men who take on receptive anal sex roll, whereas colloquially tops refers to gay men who take on an insertive anal sex roll. And versatiles are gay men who take on both insertive and receptive. So we chose to compare groups based on anal sex roll because these groups tend to differ in their gender expression. So in gender nonconformity specifically. So there is evidence that biology does affect gender expression. And so we hypothesized that these groups that differ in gender nonconformity may differ in their biological development. So to address these questions, we evaluated androgen-dependent traits between these groups. And so we looked at pubertal onset and height, for example. And we did find that gay men with a top anal sex roll present with an earlier pubertal onset, and they also tend to be taller than other gay men and heterosexual men. So next we looked at a marker of prenatal androgen exposure. So you may have already heard of this marker before. It has certainly has some controversy surrounding it. But in general, there tends to be a sex difference in this ratio. So the second digit to fourth digit the ratio tends to be smaller in men. So meaning that their ring finger is longer than their index finger, whereas females tend to have a shorter ring finger compared to their index finger. And so there's this sex difference that has been consistently shown across various samples. And there's also indications that females that are exposed to high levels of prenatal androgens, so females with conditions such as congenital adrenal hyperplasia, they show a more male typical digit ratio. Whereas males or chromosomal males with androgen insensitivity syndrome, so their androgen receptors are non-functional, they show a female typical digit ratio. So there is some evidence that this marker is related to prenatal androgen exposure. So we decided to take a look at this in relation to our groups. And interestingly, we did find a difference. So we found that tops have a more male typical digit ratio compared to bottoms, and versatiles were intermediate. And if you are familiar with this literature, then this might mean something to you. We have the Cohen's d value here, and this Cohen's d value is very close to the effect size shown in between the sexes, so between males and females. We also found that these groups differed in recalled childhood gender nonconformity, as well as measures of adulthood gender nonconformity. And here you can see that there was a significant positive correlation between measures of gender nonconformity and digit ratio. So again, these are indicating that a more female typical digit ratio is related to more gender nonconformity. So these studies indicated to us that there are likely multiple distinct biodevelopmental pathways to same-sex sexual orientation. So we next asked whether biocorrelates related to same-sex sexual orientation, whether these biocorrelates present in the same individuals or whether they would delineate separate subgroups of gay men. To get at this question, we used a statistical technique called latent profile analysis. So this is a statistical method that groups individuals who similarly vary across variables. So in this case, we're going to see how individuals who vary across biocorrelates, whether they group into subgroups or if they're all presenting together. So here, instead of using some proxy for subgroups, we're looking to see if these biocorrelates can be used to identify subgroups. So we focused on 3 well-established biocorrelates of sexual orientation. So first up, we have the fraternal birth order effect. So this is based on the finding that across cultures and worldwide, gay men tend to have more older brothers than straight men. It has been proposed that this is due to a maternal immune effect. So briefly, it is thought that with each birth of a son, the mother is at a greater likelihood of being exposed to male fetal material. And so the mother is more likely to develop an immune reaction against male antigens. Thus, with each subsequent male birth, there is an increased chance that a maternal immune response could affect the sexual differentiation of the male fetus. Next up, we have handedness. So overall, males tend to be more left-handed than women, and gay men tend to show higher rates of non-right-handedness compared to straight men. So it's a developmental trait that consistently shows this sex and sexual orientation difference. The biological processes behind handedness aren't exactly clear, but both prenatal androgens and genetics have been proposed to underlie handedness. And lastly, we used familiality of same-sex sexual orientation. So overall, gay men tend to have a higher prevalence of same-sex sexual orientation in their family compared with straight men. And this is likely related to genetic correlates of same-sex sexual orientation. So we use latent profile analysis with these 3 variables to see if they present together or in separate subgroups. And then we look to see whether these subgroups differed in gender nonconformity as well as the big 5 personality traits. So with this method, we delineated 4 groups. So our first group consisted primarily of straight men, as well as about 65% of gay men. In this group, they were low on all biocorrelates. So they're right-handed, they had low number of older brothers, and they had low incidence of same-sex sexual orientation in their families. The other 3 groups consisted primarily of gay men. So in our second profile, you can see that they had high evidence of fraternal birth order, but low on both handedness and familiality. The third profile was different from the others in that they had a high degree of non-right-handedness, so they were mostly left-handed. And the fourth group differed from the other profiles in that they had a high degree of familiarity of same-sex sexual orientation. So these results suggest that these biocorrelates are not clustering in the same individuals, but instead these biocorrelates are representing distinct subgroups of gay men. So next we look to see whether there were differences in gender nonconformity and the Big Five personality. And here I just summarized the results briefly for you. So Profile 2 was different from the other groups in that they scored high on measures of femininity and high on agreeableness, whereas Profile 3, which were high on non-right-handedness, they scored low on masculinity and low on agreeableness. And lastly, our fourth profile, that were high on familiarity, they were actually high on masculinity. So we can see here that these profiles that are differing in their biocorrelates, they're also differing in their personality and gender nonconformity traits. So to summarize this, it seems that our results are suggesting that there are subgroups of gay men who differ in their development. So we did find evidence that some might be exposed to high androgen, and we found that the biocorrelates that have been most commonly studied in this field, they're not clustering together. So this would suggest again that there are multiple distinct pathways to same-sex sexual orientation in humans. So to summarize everything that I presented to you today, I showed that I've showed some of our research looking at how androgen receptors outside of the brain can contribute to sexual differentiation of behavior. And I presented some of our ongoing work looking at how the vomeronasal nasal organ, which is outside of the brain, can contribute to sex differences in the brain and behavior. We then looked at how overexpression of androgen receptors can increase same-sex preferences in mice. And I showed some evidence to indicate that sexual orientation in humans might also result from high androgen exposure. And finally, I showed some evidence to suggest that there are likely multiple biodevelopmental pathways to sexual orientation in humans. So in the end, it's likely not only hormones or genes. There's likely multiple different pathways that affect sexual differentiation in humans. So with that, I would like to thank you all for your attention today. And I thank you again for inviting me to speak with you. I would also like to just take a moment to acknowledge all the people who have contributed to this work and the funding that supported this work. Particularly, I've highlighted Yellow Martin and Stephanie Salia, current graduate students in the lab, and lab alumni Sarah Cross, as they significantly contributed to the vomeronasal nasal organ work I presented to you today. So with that, I would happy to turn it over to Cole and answer any questions that you may have.
Yeah, thanks so much, Ashlyn. That was a great talk. Yeah, if people have questions, if they want to just throw in the chat box and say you have a question or a comment, and then we can go in order of appearance on there.
Cool, I'll try to find the chat box.
I guess I can start out with a question. So in your work with the rodents, you showed that at puberty, knockout VNO mice had low FOS activity in their brain. Is that right?
Yeah, well, we did surgical ablation, but yeah, when we surgically ablate the VNO in puberty, they are showing a decrease in FOS in response to opposite sex odors. So males in response to female odors and females in response to male odors are showing a decrease in FOS.
Okay. And is that, I know the whole idea of the talk was that androgens could be acting more non-neurally, but is the decreased activity have to do with, I remember you said it also has to do with calcium signaling, but can it also be lack of hormone action in the brain that can lead to decreased neural activity during that time?
So that's possible. We are currently doing ELISAs to look at VNO ablation mice to see if ablating the VNO is affecting gonadal hormone production. Based on like somatic measures, so like we looked at testes weight, seminal vesicle weight, bulbar cavernosus, and levator Annie weight, because they're all very dependent on androgens. And based on that, it wouldn't suggest that their circulating hormones are grossly different. But again, we're going to do ELISAs to check their blood levels. But yeah, I think, well, we know with the genetic lifelong ablation, there isn't gross differences in their gonadal hormone production either. So it seems that not having a functioning VNO is affecting the brain, right? So it's affecting how the brain's responding to these odors, these opposite sex odors.
Interesting, but it doesn't seem, even lifelong, it doesn't seem to be like cascading down like their HPG axis or anything like that and altering circulating levels or, yeah, that's interesting.
Yeah, so there was a very minor increase in females with lifelong VNO ablation. So some have like said that's probably enough to do something, but then, you know, so in my postdoc, actually, I haven't published this yet, but we did manipulate gonadal hormones in these TRPC2 knockout mice, and it didn't affect their behavior. So they're still showing decreases in aggression and increases in these sexual behaviors. So it doesn't seem to be dependent 100% on hormones.
Okay, cool.
Certainly against a lot of what the field has told us, right? For the last 60 years, we've been studying how hormones are affecting these behaviors, but certainly if we manipulate the VNO, there seems to be some great differences. So
yeah. Any questions from the audience?
looks like Levy has a question.
Oh yeah, Levi, you wanna go for it?
Sure. Can you hear me? I sometimes have trouble with my headphones, but
yeah, you're good.
Okay, great. Yeah, thanks for the talk, Ashlyn. That was super interesting. I'm just curious. Like I guess you presented some of your work on mice and then transitioned into the work on humans. I'm just wondering if, like, it seems like a lot of these studies are focusing on groups that identify as like gay men. Are there, like, in comparison, how much work is there on this hormone, gender expression, sexual orientation connection in groups that don't identify as gay men?
Yes, actually, as I was saying it, I was like, I'm kind of messed up here because the work on the latent profile analysis was actually non-heterosexual men. So it was all men who, so we used like Kinsey scale to ask about sexual orientation. So we looked at individuals who were not exclusively gay, but also were 3s and 4s on the scale. But yeah, you're right. There is a lot more research though on how gay men differ from straight men. There is very little about other individuals on the scale, so bisexual or between bisexual and gay men. There's also very little research on individuals who identify as non-binary, which again, there are a lot of indications that biology do affect gender expression, so that certainly would be an interesting area of study. Doug Vanderland, if you're interested in that kind of work, he does a lot of work looking at third genders in other cultures. So he's looked at the Fa'afafine in Samoa, and he's currently publishing a lot of work on Thailand. So there's still a lot of unknowns for sure in that respect. Did I answer your question? Or?
Yes, thank you. Yeah, I was just kind of striking the sort of the one group that seemed to be studied quite a bit.
Yeah, no, it's true. It's kind of like in experiments, it's easier, right? Like you're more likely to get individuals identifying as either gay, bi, or straight, but there's certainly a lot of variability in between those categories. And yeah, a lot of work can be done there still. So.
Yeah, thank you.
No problem.
I have a question.
Yeah, go for it, Anne.
Okay, so with the familiality, does anybody look very much at how much might be hormonal similarities in family members and how much is just differences in tolerance or acceptance of alternate sexual preferences in families?
Yes, so that's something that we were definitely discussing when we were doing this research, because it's, how are you supposed to know if your uncle or your cousins or whatnot are gay, or otherwise bisexual, if your family is very conservative, right? So if your family is very conservative, very religious, against same-sex preference or sexual orientation, there probably is an underreporting, right? So that's certainly a limitation of this research. But yeah, it's just a first step, I guess, to kind of try to understand whether these things that are so often studied are grouping together or not. But
I thought it was really interesting because I've heard of the fraternal birth order one and thought that it only applied to a segment of same-sex preferences, and that's what you're showing. So that's interesting.
Yeah, and what was interesting about that is those that studied the fraternal birth order effect, they have said for years that it likely only accounts for about 14 to 15% of same-sex sexual orientation among men. And that's actually really close to what we found. So it was 14.15% of our sample that fell into that group. So there was definitely a lot of links like that were interesting.
But even if it's just a first step, I mean, it's good you're thinking about what could be going on in terms of hormones or social things.
Yeah, I know that's the hard part with humans, right? It's so much easier.
Yep.
Yeah.
Great. Any other questions from anyone in the audience? I have one more. I'm not sure. I don't know if this is a good measure, but you say that within same-sex orientation in human, that androgen levels can vary. There's no consistent levels between individuals. Some have high, some have low levels of androgen. Is that right? Is there any work that's showing, is there like an androgen to estrogen ratio that's a good tell as well? Or is that, I'm just spitballing here. I don't know. Yeah.
Yeah, like it's really hard to get at the hormones in humans because like we've talked about, right, it's mostly these prenatal hormones that we would want to be studying. And so we're trying to find markers that we can study that are related to these prenatal hormones, but none of them are great. And, we don't know, right? So one of the things relating to your question is like the 2D/4D ratio. So everyone does talk about it, and I'm obviously do it as well. Everyone talks about it as a marker of prenatal androgens, but there is indications that both androgens and estrogens are mediating this digit ratio difference. So for example, if you look in the rodent literature, and there's more research coming out every day. So I was reading even a research in a non-human primate with digit ratios, and it seems that both androgen receptor and estrogen receptor affect digit ratio lengths. And so does cortisol. So there's a lot of hormones that are affecting it, right? So yeah, no, it's a very good question and something that I'd be certainly interested in trying to get at, but it's challenging in humans for sure.
Yeah, I believe that.
Definitely a good question though.
All right, well, if we have no other questions, I guess we could just say thank you for your seminar today, Ashlyn, and thanks for coming over from the psychology department and presenting in biology. I think that's really cool.
No,
Yeah.
this is really great. Thank you for having me. It's great to share some of our new research too in there, and thank you for your questions and having me here.
Perfect. Yeah, and if anyone, I'm sure people can get in touch with Ashlyn if they have any questions after. Yeah, so thanks for coming, and yeah, we look forward to see the work that's going to come out of your lab, since you're relatively new here at MUN. So it's exciting to see what you're going to do to pave the way here at Memorial. So yeah.
Thank you. Thank you. Yeah, I'm excited. I have 3 grad students now, and they're all working very hard on this stuff. So I think we'll have a lot of interesting stuff coming out in the next year or so.
Cool. All right, cool. Thanks, Ashlyn. We'll see you around the building, I guess, maybe.
Sounds good. Take care.
All right, bye.